Welcome to the introduction to the Skeleton Lab. The first thing we want to do is take a look at what you need to do in order to prepare yourself for the first of the two skeleton labs. What you want to do in preparation for next week, and these are all items that you can be accomplishing during this week, is make sure that you are familiar with all the bones in figure 715, page 207 in the Holes textbook. And this is, of course, going to be in the 11th edition of the textbook. For you folks using Merib, there's a very similar illustration. What it is are just the major bones of the skeleton. You should know your femur, your radius, your ulna, all the major bones of the skeleton before you even walk into class. And most of you come into the class already having a knowledge of those bones, so this should be a very minimal study for the majority of you. You also want to make sure that you know all the bone features in table 7.4 on page 208. This introduces you to what's the difference between a fossa, a caruncle, a ridge, a foramen, a crest. What are those structures that we're going to use to describe different features of the bone? Because a lot of what we're doing in this lab is actually not just looking at the names of the major bones, but the features on those bones. Once again, what's a foramen, what's a fossa, what's a crest, what's a tubercle, what's a trochanter, what's a condyle. All those are features that you need to have knowledge of before you walk into class for our first lab session dealing with the skeleton, because we're interested in these features because it's where bones articulate together, where they come together and form joints. It's where blood vessels move through. It's where arteries and veins move through, blood vessels, nerves, etc. So make sure that you're familiar with those before you get into class. Also know the bones of the skull in figure 719, page 210. And once again, Merib will have a very similar illustration. Know the major bones of the skull. You don't have to know the little features of the bones, just know the bones. So we're talking temporal, parietal, frontal, etc. Those that are listed in your SPOs. Don't worry about the features of the bones, just know the major bone plates, because this is where we're going to be starting. We're going to be starting on the axial skeleton, which of course incorporates the skull and the vertebrae. So that's our first lab. So make sure that you have that taken care of before you get to class. Then what you want to do is watch the Skeletal Lab intro, which of course is what you're watching right now, and this will give you some additional background for that first lab. And then begin working on your SPOs using the APR CD, the Anatomy Physiology Revealed CD, and the Biology Learning Center. There's absolutely no reason for you to wait to begin your study on this unit until you get to the first skeleton lab. In fact, don't do that. Because if you count how many SPOs you have, divide by the number of days remaining till the exam, leaving 4 or 5 days to review, you've got to get started. So you really need to walk into that first skeleton lab having a lot of this under your belt already. And you can certainly accomplish that with APR and the BLC. Now, as we get into the lab and working in the first lab, there's some guidelines you want to follow. Stick with a bone until you know it. Resist the temptation to move on to another bone because one of them is giving you a problem. So if the femur, for instance, is giving you a problem, don't put it back in the drawer and move on to the tibia. Stick with it until you know it. Otherwise, you're going to be treading water and really getting nowhere. Please be very careful with our bones. Most of our bones are still natural bone skeletons, which we can no longer get. We just can't get human skeletons on the international market anymore. Everything now is a plastic cast or reproduction. So please be very careful with our natural bones because we're not going to be able to replace those any longer. Don't use pencils or pens to point at our bones. Use pipe cleaners or some of the long wooden Q-tips because we don't want our bones marked. We don't want them damaged. So please be very cognizant of that. I know it's kind of natural to take your pen or pencil in your hand and then point with it, but please don't do that with our bones or any of the models throughout either of the 2 semesters. Also, return bones to their proper location. You'll notice that all the bones coming out of the drawers are properly labeled. Make sure that you take the time to A, figure out what bone you have, and B, figure out what drawer it's supposed to go back into. Correspondingly, when you take a bone out of the drawer, don't assume that it was put back correctly. Make sure that, in fact, you're looking at the bone that you're after and don't start trying to learn femur features on a humerus, for instance. So take a few minutes to make sure everything's going back either on the counter or in the drawer where it came from. Everything is labeled, so please check the labels. Be sure you know how the bone fits into the articulated skeleton, because virtually none of your patients that you folks are going to see are ever going to come disarticulated, which means put together. We have each of the each of the four people sitting at a lab table will be sharing one fully articulated skeleton, and we have drawers and drawers of disarticulated skeletons that have been taken apart. But since all of your patients are going to come hopefully fully articulated, even though you may have just a femur in your hand, make sure you know how the femur fits into the articulated skeleton. And that's going to help you understand how the joints work. So as you're studying a bone, roll over the skeleton, the articulated skeleton next to you, and take a look and understand how the two are connected. You should be able to distinguish between right and left bones for the following bones: the femur, the humerus, the radius, the ulna, the scapula, the tibia, the coxal bones, and the coccyx and the sacrum. And you should know also anterior versus posterior for the coccyx and the sacrum. The reason for doing this is that it forces you to learn certain features of the bones as well as understand how it fits into the joint. So by having you understand is it right or is it left, you'll understand how it articulates into the joint, and you'll learn the bone a heck of a lot better that way. Now when we take a look at long bone anatomy is something that we're going to study both in lecture and in lab, but we know that all long bones, and as the name implies, longer than they are wide. So bones like the femur, the radius, the ulna, the tibia, the fibula are long bones. They have kind of a characteristic anatomy. They're composed of an area of compact bone, which you see indicated right here by the cursor, and we'll take a look at another illustration of that in a second. Here's a layer of compact bone right here, as well as spongy bone on the inside. But let's take a look at the gross anatomy first. The main stem or the trunk of the bone is called the diaphysis. So, not including the two ends, this trunk of the bone is called the diaphysis. The two ends of a long bone had what's called an epiphysis, not epiphany, but an epiphysis. There's the proximal, which means it's closer to the trunk of the body, and the distal epiphysis. You'll talk about in lecture as to how bones are actually formed, and this idea or this knowledge of what's a diaphysis and what's an epiphysis are kind of critical to understanding how bones are formed and also how they're repaired, both naturally and artificially. You're also going to see the center of the bone has an area here called the medulla. Medulla just means center, so this is called the medullary cavity. It's the cavity in the center. And in the living bone, this is where you're going to find yellow marrow, which is fat. In the prepartum skeleton, just before birth, prepartum means before parturition. Parturition means birth. So prepartum is before birth, postpartum is after birth. In the prepartum skeleton, you're going to find the medullary cavity full of what's called red marrow. And this is actually a blood-forming tissue. But after birth, that red marrow is replaced by yellow marrow in the medulla. However, the spongy bone that we see right here, the red marrow persists in the postpartum skeleton. So after birth, we still maintain the red marrow in the spongy bone, and that's actually where we produce our red blood cells as well as our white blood cells. In lecture, you'll spend some time looking at these structures here called the epiphyseal disc. Over here on the left-hand side, we actually have a fresh bone that's been cut to expose some of this yellow marrow. And ask your instructor, in fact, of course, that's going to be me unless you visit another lab, to make sure that they show you the frozen cow bones out of the back freezer. I've had them cut longitudinally and in cross-section so you can see all these features, including the structure right here called the perio, which means around, and oste, which means bone. Together, the periosteum. It's a tissue that actually encompasses the bone. Anytime you see a bone that your dog has been chewing on, this layer is missing. It decomposes and falls off. Periosteum is very important because the bone-forming cells reside here, and as well as the tendons and ligaments attached to this layer. You'll spend more time with that in lecture once again, but make sure that when you're looking at the frozen cow bones that you look at this feature because it'll be absent on the dried bones. Let's now take a look at a long bone in greater detail. So here we have the compact bone. What we're going to do is take a little slice here of compact bone and bring it out here. We can see some of the microscopic structure. You'll notice that compact bone is comprised of a whole bunch of these cylindrical structures called osteons, which just means bone unit. Osteo means bone. These used to actually be called Haversian systems, named after the person who first described them, but we're now calling them osteons. And you notice a whole series of these osteons make up the compact bone. The spongy bone over here is made out of a different structure called trabeculae, which is a fishnet spiderweb-like pattern. So long bones are made out of spongy bone with a structure called trabeculate and compact bone with structures called osteons. Let's take a look at the osteon. The osteon has going through the center these blood vessels. You're always going to have a little arterial, a little venule, and also, illustrated in yellow here as a matter of fact, you're going to have nerves. Almost everywhere in the body, when you have blood vessels, you have a nerve connected to them. So each osteon is comprised of these concentric rings of bone cells and something called the matrix we'll talk about in a second, as well as blood vessels. The center, this canal through the osteon, is called the osteonic canal, and this is where the blood vessels and the nerves actually move through vertically down the length of the bone. But they have to move horizontally also. So these canals that perforate through the bone that allow the blood vessels and nerves to connect with each other, and that's called the perforating canal. You can once again over here see the periosteum. Okay, now we'll come back and take a look at this in just a second in a scanning electron micrograph. But before we do that, let's look at the illustration. If we take a little section right here and we enlarge it, we can see the bone cells in here, and these bone cells are called osteocytes. Osteo, bone, cyte, cell. These are mature bone cells. You'll learn about other types in lecture called osteoblasts, and also you'll look at osteoclast in lecture. But right now, this is an osteocyte. These are mature bone cells, and they live inside these little corridors here. Now remember, bone is a connective tissue, and by definition, a connective tissue is comprised of cells, obviously, like all tissues, as well as a matrix. The matrix is composed of fibers as well as a ground substance. The fibers found in bone are going to be elastic fibers and collagen fibers. The ground substance is going to be this calcium phosphate salt. So in bone, which is kind of unique, the cells are actually entombed. They're trapped by this calcium phosphate ground substance. They actually have secreted around them. But as we know, in any tissue, cells have to communicate with the cells next to them. So how do you do that when you're encased in a rigid calcium phosphate? So these little corridors right here, called canaliculus is plural, and canaliculus is singular, and canaliculi would be plural. You can see how little extensions of the membrane from one cell come in contact with the membrane from the other. And in this manner, the cells are actually able to exchange nutrients, waste products, communicate with each other. So if we come back here and look at this illustration up here, we can see these little canals going between each of the lacunae where the living cell is housed. Now, when you look at preparations, you're actually not going to see these cells. In fact, let's go to the next illustration and I'll tell you why. On the upper left-hand side here is an artist's rendition of what the micrograph looks like down here on the right-hand side. They always label, once again, here's an osteon, and here are the lacunae. They always lead you to believe that you're looking at the cells that are in here. In fact, look at this. They've even labeled it as osteocyte in the lacunae. It's not there. It's missing. Here's what the cell would actually look like. But when we prepare these slides for you, what we actually have to do is cook the bone first in the oven to recrystallize the calcium phosphate so it'll accept the biological stains, the dyes that we use. And in the process, obviously in the oven, you destroy the cell. So even though they always label it as osteocyte, there's no osteocyte there. You're not going to see it. All that remains is just the cavity. But you can see these little lines here, these fine lines, which are the canaliculi, which connect one osteocyte to the next. And here, of course, is the osteonic canal, where the blood vessels and nerves would be in the actual living bone. Here's a scanning electron micrograph of an osteon. Everything is labeled there for you. And once again, you can notice that there are actually no cells in the lacunae. Okay, the goal today is going to be to take a look at, for the first skeleton lab, is going to take a look at the axial skeleton. Everything illustrated here in orange is the axial skeleton, and this includes the skull, the vertebrae, the sternum, and the ribs, and a portion of the pelvis called the sacrum. Okay, so that's the goal for today. Next lab, we're going to continue on to the appendicular skeleton. The appendicular skeleton is illustrated here in brown. But for students who are prepared, you should be able to blow through the axial skeleton today and actually get started on the appendicular skeleton. Take a look at this reference out of your Holes textbook. There's a very similar illustration in the Merib textbook, so you understand the difference between a long bone, a flat bone, a sesamoid bone, and a regular bone, because those are terms that will become important today. Here are the bone markings that I made reference to before. Make sure you're familiar with these bone markings, because a lot of your work here is going to be actually studying the bone markings, not so much the main bones, because most you come into the class already knowing the major bones. Okay, so that's where I'm going to leave you, and we'll have you guys walk into the lab good to go. Take care.